Year 13, welcome back. We're going to carry on this week with our notes on the first topic. You can see we're on to part C now, transport in plants. We've done the lungs and different things last week. So anyway, we'll get partway through this, just a few pages in to try and get some of the structures understood. The xylem, the phloem, are the two that we need to know about, and then next week, or rather we'll probably push it back a few weeks, because next week we're proposing a bit of a catch-up week, then half term of course, just to give everybody a break, and then the week back towards the end of February, then we'll carry on with how water and minerals and sugar actually move through these structures. Okay, so you should have this booklet anyway available to you, I hope, which 2.1c. Okay, so I'll be dipping in and out of this, and I've also got it then on this PowerPoint. So we'll start with this. Okay, transport in plants, structure of the xylem and phloem. The xylem, of course, you should be familiar with from GCSE, in that it is the system where water and minerals get transported around the plants. So again, most of that will be refresh. But we start here with the main, I suppose, tissues within a plant. Okay, and this is straight at the start of your notes there. You can see plant tissues in relation to water and ion transport and translocation. So there are four main plant tissues here, and there they are listed. You've got the epidermis, endodermis, xylem, and phloem. Okay, the epidermis really is just the outside layer, okay, protecting it from the environment and anything on the outside of the structure. Endodermis then is pretty much the internals, and then your xylem carries the water and minerals, and your phloem, which you've heard of, I'm sure, but maybe don't know an awful lot about, is the kind of sugar pipe. Okay, it's all that sucrose that a plant makes from photosynthesis, and how does it get around the structures. Okay, so those are the four that go into page 1 there. Then ask you to sketch some diagrams. So I have these up for you. Okay, I'll put them a little bit bigger, so as if you're going to try and annotate them and so on, you have it all there. But this is at the bottom of 1, where you have a cross section of a root. Okay, and so what is very distinctive about a root is the X or cross shaped xylem. Okay, that's the thing to look for. All right, up the middle of every root of a plant has got this cross or X-shaped xylem structure. So what we're going to learn next week is how does water get from here in the outside, okay, through these root hair cells, across this cortex, in through this pericycle and endodermis, and then into this xylem. And then it's going to go up, okay, towards the leaves. Now you'll notice also between the arms of this cross, then you've got the phloems. So there are multiple phloems, okay, usually the 4 that are interspersed between those extending arms there. Now that whole thing, okay, is called a vascular bundle, and that might be a GCSE term you remember. So it's basically the sections, the discrete sections that are carrying the fluids, and in this case the stele is the whole thing, including the encapsulated endodermis and pericycle as well. So those are just little bits of structures, and you'll recognize the outside part, root hair cells, and that's the epidermis. Okay, so you have the endodermis in here, epidermis there, packing tissue called the cortex, and then this is the bit we're interested in. So you can pause it here, and you can take a sketch of that on page 1. Okay, page 2, you're asked for 2 diagrams. You haven't got an awful lot of space, so maybe you want to split it down the middle or find some other room. But what you've got here is a cross section of the stem, and you'll notice it is different, okay, distinctively so. There's no middle, okay, stele here at all. Vascular bundles are actually around the outside, and there's multiple vascular bundles, not just one central one. Okay, so they look like little kind of egg-shaped things. You'll notice there's a line separating 2 parts to it. Okay, on the outside, the dark brown in this picture is the phloem, and on the inside is the lighter brown xylem. So they're bundled together, but they are literally scattered around in a concentric circle there around the outside. Okay, running parallel to the epidermis again. This time there's no root hair cells because there's no absorption through the stem. There you have a cortex again. All right, and lots of other fancy words that sometimes come up in different textbooks here. Okay, some plant packing tissue, collenchyma, parenchyma, sclerenchyma, fancy words that say if you read, don't panic about. They used to be on your syllabus, but they've been removed. But they're good words to have in your vocabulary. Okay, so take a sketch of that as a cross section through a stem. One way it helps me remember what's on the outside is if you remember things like green fly, little aphids come to suck out the juices of rose plants or whatever, they will land on the outside, clearly epidermis. They will stick their little nozzles, their proboscis, through, and they are trying to hit the phloem to basically get all the good sugary fluid. And so, knowing that that's what they're looking for, that's on the outside, okay, of this vascular bundle. So phloem outermost, xylem innermost. Okay, third diagram then to be squeezed on the page 2 is a cross-sectional leaf, and this one you're most familiar with. You've done it since first year, so you've got your upper epidermis, lower, you've got your mesophyll, palisade and spongy through this bulk of the middle, but scattered throughout it then you've got these vascular bundles. Okay, you've a main midrib. If you look at a leaf, you'll notice that there's a big main vein running up the middle, mostly xylem with a wee bit of phloem, and then you've got side branches, okay, veins that will extend throughout the mesophyll. And don't forget, what they're trying to do is bring the water and the minerals, okay, to these photosynthetic tissues, okay, particularly the palisade ones, and at the same time they're collecting all the sugar, okay, that has been made there, and putting it back into the phloem to be then run throughout the plant. Okay, some goes up to the developing buds and flowers, to other growing parts, and others go down to the roots to maybe be stored into starch and you know, in the likes of a potato and so on. Okay, so anyway, that picture with all its labels, please, goes on page 2. Okay, went to the xylem tissue then. So the first question, what is a tissue? Just a quick reminder, of course, a group of cells working together to perform a particular function. Okay, so similar cells packed in with a job to do. So you can see in your notes there that your xylem is a tissue. Okay, its main job is to transport water and ions, okay, around the plant. Highly specialized, composed there of chains of cells with no cell contents, no cytoplasm, nucleus, or other organelles. Okay, well worth highlighting some of that, I would say, in your notes. And let me just see if we can do that. I'm going to add a little bit of drawing onto this, mainly because I think I can. And we'll do highlight. Okay, and it says here, okay, so highly specialized, so no cell contents. Now I can hopefully get this to work. No, it's not for some reason. Why is it not working? I don't know. We'll just move on. Nothing to erase there. But these are the things you could highlight, okay, and there we go. No cell contents, nucleus, or other organelles. Okay. The other big thing is this: these are dead cells. Okay, they obviously start alive in order to get put into place, but they go through a kind of sacrificing of themselves, and at the end of the day, they are dead. There's nothing living within them. Okay, there's no requirement for respiration. They basically become hollow tubes that allow water and minerals to come travel through. Okay, so they form a continuous tube with holes basically at the end walls. Now here's a little question in your notes: why? It's to allow basically a continuous flow of water. Okay, there's no need to go from cell to cell to cell. You remember from water way back that it has a cohesive property. Okay, there's a stickiness between every water molecule. Well, that only is functional if they're actually in contact, and once you have a cell membrane and wall to get through, you lose that. So therefore the xylem is one complete tube, okay, from bottom to top, and each cell that builds it then gets its kind of end hollowed out, and so it becomes a big long tube. So there's a short answer to this: to transport water, okay, swiftly and continuously from the roots to the leaves, of course, for the primary function of photosynthesis. Okay, and we will learn more about uses of water as it goes up through the plant. Lignin. This is the main thing that gives it its strength. Okay, it's this woody material. Okay, it's basically, when you look at wood, before you burn it in the fire, this tough kind of waterproof material. Okay, and its job is to provide strength. Okay, so as it doesn't collapse, and as you're going to learn, there's pressure differences that exist within this continuous tube. It's equivalent to like a sucking sort of action from above, and so the walls would collapse on themselves just like a straw would if you were to suck on it too too hard and the sides just give way. So this lignin prevents that. It keeps it kind of hollow with a lumen, a space for the water to go up, and of course makes it waterproof. It means that water cannot escape unless it's given gaps in places to do that. Okay, and in order to allow water to go out at certain places, then there's pits, little holes that allow for sideways or lateral movement of water. Okay, so those are some of the main things within the xylem. Let's go back to the PowerPoint and have a look. Okay, you'll discover that xylem tissue is different types of cells working together. It's got a protoxylem and a metaxylem. Okay, so the whole structure is divided into two, and it really depends on their characteristics and when they get made. So proto comes first. Okay, and meta then is the more mature one. So that's kind of a good way to remember it. Here's just a quick cross section of both of the vessels. The xylem here on the left, you can see it's continuous tube with dead lignified cell walls up the side. The only thing missing maybe is that there's no wee pits, lateral movement out of this particular picture, but you notice they're non-living, only one way. Water has to go from beneath up to the top. No walls between cells, and its contents are water and minerals. The phloem, as we'll come to shortly, these cells are alive. Okay, but they need support. They have these sieve end plates called, okay, which are basically like a sieve, okay, a colander. They allow things through, but they have to also provide a two-way system here. So within the same pipe, you have sugars that are going upwards towards maybe developing buds, flowers, and growing structures, and then also some going down towards roots and other places down beneath. So this is the organic molecules, primarily sucrose. So the vessel elements, this is your xylem. So this really ties in with page 2, as we've already highlighted. Elongated tubes end to end, okay, for a continuous flow of water. Dead cells, lignified, makes it very hard, waterproof. They have an empty space inside, an empty lumen, contains pits, places where there isn't any lignin, so as water can move out. Okay, this is where originally it would have been plasmodesmata. You remember that structure from cells back before Christmas? These were the gaps between cells that allowed for swift transport rather than having to go through a wall and a membrane and slow things up. So this is basically glorified plasmodesmata. End walls break down completely to form these continuous tubes. Now, there are four main patterns, though, that they can form, and depending on their age. As when a plant is growing, like the very first picture in this PowerPoint, you saw there was a tiny little shoot coming up between some cracks. It has to be able to grow and stretch, and so any xylem that's formed has to be able to bend and twist and move and change shape. And so, therefore, the lignification is usually allowing for that. Okay, it's going to be spiral, like this one here. Okay, or it's going to be annular, ring-like one, and that allows for flexibility while it's going through these growth stages. And this would be your protoxylem, okay, your first two main early on sort of xylem lignified patterns. Then when it gets more mature and obviously it's not going to change shape very much and not going to stretch very much, then it becomes reticulated and pitted. And reticulate is just starting to fill in the gaps. It starts to kind of bulk out a little bit and become more secure. And the pitted is almost everything's lignified except to say for these lateral little channels that allow it to go left and right in lateral movements. Now the picture you get to draw then is this one at the bottom of page 2. There's your spiral and annular. If you give them a quick sketch, okay, ring-like compared to these kind of obviously like a helix spring shape, and then on the metaxylem, so the more mature one. So as a plant it gets that stage where it's not going to change in height or size too much, then it becomes more reticulated, okay, which is more stripy, okay, almost camouflagey sort of stripes there, whereas the pitted is everything is done bar these little lateral holes. Okay, so put that in page 2 at the bottom, fit in as best you can. These are some pictures just of it. You can get to see. Okay, here we have quite obvious sort of ring or spiral shapes down this left-hand side in particular. This arguably might be a wee bit more, stripy here. Could be a bit more pitted. You don't see the pits too much. This is probably a phloem here beside it, but there's definitely a kind of a protoxylem effect of spiral or ring type one there. Again here, you can see that, and that's very distinctive of a xylem. There you go. right? Your annular, your rings, and again here, right? So that's quite immature xylem. So that's going through early days of a plant, and you can see it's allowing for a little bit of flexibility as it has to stretch and it has to bend maybe between rocks and different things in order to grow and establish itself. Okay, top of three then. There are two main types, as we've said, proto and meta. So highlight these in your notes, 1 and 2. The proto is the first to develop. So pro just meaning kind of before in the region of elongation just behind the root and shoot. You might remember a little bit of that from GCSE. Lignin is added in rings or spirals to form those very distinctive patterns. Okay, then as time goes by and these parts of these plant structures don't need to change much anymore, then it's kind of reinforced with more lignin to become metaxylem, and that's, I say, your other two types that you've seen. Okay. Right, so we're into the second one. This is the phloem. You can see here on page 3. Okay, phloem tissues consist of a number of cell types. The cells primarily concerned are called sieve tube elements. These are aligned end to end and form continuous row of sieve tubes. But unlike the xylem, they do not have end walls, or they do, sorry, have end walls, and these are perforated with sieve pores to form sieve plates. Okay, so these are the main cells. Just to have, it's not really listed, I don't think, in your notes anywhere. It may be good to scribble just these down at the side of that on page 3. The sieve elements, companion cells. Now, this is one of these fancy packing tissues I'd said that are found within plants. Parenchyma is how you pronounce that. And then these little fibers as well. So there's a picture then asked for on page 3. If you've got a textbook, you'll find it there, but I've taken a photo for you, and this is it. Okay, so here we have one cell above, another cell beneath, and the central cell is one sieve tube element. It's got perforations in its top and bottom, and there's it looking down. Okay, you can see it is very much like a sieve, a colander, so that's a sieve plate, and that allows a continuous flow, okay, between cells, so you're not having to cross the membrane. Now, each sieve element, though, is got, well, at least every now and again, has got a companion cell. So it's this kind of body on the side. You notice again plasmodesmata between them, so as you can have an exchange of materials and such like. Now, this they're all living cells. There's nothing lignified here. Okay, so lignin does kill a cell. It stops obviously water coming and going since it's waterproof. So there's no lignification. Okay, but there is a need for, as it gives you a hint with all these mitochondria, there's a need for respiration. Okay, transporting sugars around the plant is energy rich. It requires a lot of respiration to happen. Okay, so a plant will expend a fair bit of its food resources on simply translocating or moving sugars around. So there you have companion cell, which is the energy source basically. So mitochondria are packed in there, lots of ATP will be processed in order to allow sugars to move. And you know, there's a number of reasons, as we'll see. You've got a prominent nucleus, but there's a two-way system going on here. Sugars can go up and down the phloem at the same time, and so therefore that's not going to happen without possibly, you know, protein again carriers, machines, and so on, like we saw when we looked at cells being able to grab and pull things in the direction that they need to go. So that picture goes on page 3. Here's just another little cross section of it, okay, giving you appreciation. You can see the sieve tube element. There you have the sieve plate at the end. Here we have smooth endoplasmic reticulum. Any sort of cell contents are quite often squeezed to the outside, okay, allowing as clear a channel down through the middle as possible. You notice a couple of little things, thickened primary wall. Okay, there's a little bit of pressure in here, so it has to resist collapse or kind of expansion, and so therefore it does have a slightly thickened wall. You'll notice then the companion cell has got all the usual things, but it's very energy rich with lots of mitochondria. You're going to have lateral sieve areas too, a bit like the pits in the xylem, being able to allow sugar to access this, okay, coming from leaves and able to go therefore up or down the plant according to where the demand is. Again, some more. Notice again, no lignification. There's a sieve plate. Okay, you've got a few little pits there, lateral pits. These are companion cells attached to the side. Sieve elements then. Okay, this is part of your notes from 3 into 4. You can just go through and highlight living cells have a cell wall membrane cytoplasm containing ER and mitochondria, no nucleus or ribosomes within the main sieve elements. You do get them within the companion cell, and I say there's big pores at the end of each cell, top and bottom, for the sieve plate. These join together end to end to form a continuous column called a sieve tube. And there's a cross section of one. You see clear gaps between them. Okay, again allowing a continuous flow without hindrance. The companion cells then, and this is over on page 4, found beside sieve elements, have a large number of mitochondria and ribosomes, numerous plasmodesmata between them. So these are kind of the workhouses that are going to provide energy and other molecules needed if there's transport proteins needing to be made to get its sugars pulled along, then the ribosomes are doing that. Okay, and that's going to take you to the halfway down page 4. Okay, at that point we're going to leave the notes because the next bit we're going to leave to next week, but could you then go on over to page 11, sorry 10 first. If you go there down at the bottom, 10 is just a little bit about the phloem and how things move, so we'll do that now. It's called translocation. Okay, and so translocation, let me come off this and find out where we are with this here. Okay, you've got all that, and I say we've moved on down. We'll come to this stuff next week, and here we are here. Okay, translocation describes the movement of organic substances, and the key is this. So that's anything that's been made through photosynthesis. It's going to be sucrose primarily. Okay, remember that's a glucose and a fructose stuck together. It's energy rich. It kind of for every molecule transported, it's kind of got more energy contained within it, so it's very economic from that. It doesn't have the same osmotic kind of influence on the fluids within a plant as maybe glucose would, so it's another reason why it's done in that form rather than just pure glucose. Okay, so it's transported in the phloem and it goes to growing regions and roots, so it means up and down. It requires energy. Companion cells will give you that, and it's also a two-way system. Now, I then ask for evidence for energy. What is the evidence that energy is required for this translocation sugar movement, and then the evidence for two-way movement? All right. Now, you can see it's referred to your textbook. If I go back to my PowerPoint, this is what I've put up for you here, so you can pause it here and basically take out the information you need. Okay, companion cells and metabolic inhibitors such as cyanide stop the translocation, so that's proof that respiration is a vital process. Okay, so you can take out some information. And in the two-way system, you can see there's a little experiment where they tracked carbon-14, okay, through the stems and so on, and they were able to find that it was going up and down, and you can read that yourself. There's a bit of mass flow. Okay, it's localized buildup of sucrose source, and it creates a hydrostatic gradient. All right, basically from source to sink, so where there's lots to where there's little. Okay, and where sucrose levels are lower, for example, in the roots, because it gets built up into starch, for example. Remember our potato kind of experiment, or sometimes it gets used up in growing regions as well. So anyway, you can pause it there, take in that, and then just to finish this little bit, this was this experiment about the carbon-14, and you can just see where they took cuts and what they went to find, and wherever they took the phloem from. Okay, ringed bits around the outside. Remember, there are phloems on the outside of the stem when it comes to vascular bundles. So if they're cut, then basically no carbon-14 was able to get up. Okay, so it basically originated from this leaf and would have gone up. Okay, so it would have gone up this way in this experiment, therefore it was recorded there. It also went down, so it was recorded in both. In this one, it would have gone up, except it hit this region where there was basically no longer access to get above it, so hence that's why there's none found there, but yet it went down. So it was to prove that it didn't come from necessarily these leaves. Okay, the carbon-14, the only source was this leaf here within this bag. Okay, so we can fill those in up to page 11, and I say we'll go back and do how water is transported through plants, which is pretty much here. Okay, next time. Okay. All right. I'll set you some questions just on that as well, and there'll be another wee quiz, and we'll be in touch. I hope next week to maybe do a wee live feed. I'll get a time set aside, and we can just touch base and check how everybody's doing. Okay. All right. Let me see how you end it. Right. Bye.